Alright, my bio nerds, you asked me 3 times to film this lab, and I decided I will. I tried on Friday, but kept getting interrupted. So it's Sunday, and yeah, I should be out running, but instead I'm in here. So what I'm going to do today is try to give you a good overview of the setup for this lab. We're not going to get the results today because I just placed the seeds in for germinating. So we're going to have to just kind of go through the procedure. Again, we're going to do this on Wednesday, and this lab can be good, but it cannot be so good. So as you today, I am joined by my friend Leo here. He's going to make sure that I stay on track, and I'm also wearing my safety goggles because I got some grief for not following proper safety procedure, even though I'm using peas and distilled water. But hey, everybody's got to complain about something. So hopefully for Wednesday you'll read through this lab, and I'm pretty sure about 75% of you won't. But we can only hope. But as you can see, the title of this lab, Carolina Cell Respiration AP Bio. Not a big fan of cookbook labs, but this one does allow for a little inquiry. But again, the biggest pain is getting it set up, and so that's what we're going to take you through today. The first thing I want you to see is I do have a black tray right here. Black tray. I've got my thermometer. And then the directions say to place pieces of white paper in the bottom. The white paper tends to float, so I'm using these dry erase boards that we use to answer questions on sometimes. So I'm going to place those into the bottom. I'm going to take my thermometer here. It's about 24 degrees Celsius right now in the classroom. I'm going to lay that in here just like this, and I'll show you this after. I got me some water. Water's important, so I'm going to just kind of dump this in. I'm going to try to get it filled up to a point where my respometers will actually be able to be submerged. And it's important that the respometers are being submerged because that's how we're going to calibrate them, so to speak. Probably should have had a little more water. Oh, guess what I got? Got more water. So we're just going to keep adding more H2O. And we're going to fill it about halfway. That should be good. We're going to need this water for later. So I've got my thermometers in there. My thermometer, excuse me. It's kind of floating in there. So here's the aerial view of the setup. Alright, so we're back. I got the tray set up. Now we got to set up our peas and the beads and the respometers. If you're looking at part 2, the directions here, and again, they're going to be backwards because of this. We got to put 25 milliliters of water into a graduated tube. So I've already got some in there. I'm going to keep adding it until it's at 25. There's my 25. You can't really see it. Now, this is where things are a little different. I was supposed to put germinating seeds in there. Unfortunately, Mr. Montessori didn't plan well as usual. So we're going to pretend these are germinating seeds. But again, this whole point of this video is just for the process. We put those in there. Lost one. Lost 2 actually. And I'm sure you're going to do the same thing because none of us are perfect at these labs. But as you can see by placing in my germinating seeds, it displays it till about 30. So as the directions say, you would write down 30 mils, and that's equivalent volume of peas. Record the volume. You're going to remove the peas and then put them on a paper towel. So we're going to do that real quick like. We want to get off all the excess water. So I do that. So that's the first set of peas, and then I would set those off and label those peas germinating. The second step, we're going to fill up our graduated cylinder here up to 25 mils. And again, now we're going to place our peas that are non-germinating into the water. Hopefully you guys can see this. And we got one little floater in there. These are at about 28 mils. So even though I've got the germinating, non-germinating, these are still about 28. So you would record that information, as you can see there. And then the same thing, we're going to remove the dormant peas. Sorry you guys are getting a picture of my armpit, but it happens. Place those onto a paper towel. Voila. Next, we are going to again fill up a graduated cylinder with 25 mils. It's 25. And then we're going to add enough beads. I got these red beads here. Red beads. Look at the red beads. We're going to put these in to equal the amount of the germinating peas, which was about 30. So there's my first, and I'm a little over. So I'm probably going to I'm about at 31. So what I'll do is I'll take these beads out, and then I'll calibrate them until I know I've got that the beads volume will equal that of the germinating. So I'm going to do that now and get this all set up for you. So let me get myself situated, and I'll be back in a second. Alright, I'm back. I had to move Leo here. He was kind of upset that he wasn't getting a starring role, and he kind of growled at me a little bit. No, not really, but anyways, so I've gotten all my peas back into these cups, germinating, non-germinating, or dormant, and then I've got the beads here. So we got those set up. But what we got to do now is set up our, we got to prepare our samples. So what I've already done ahead of time is prepared a respometer. So here is the glass tube, glass vial. It's got a metal at the bottom. Here is the cork with the pipette in it, and it'll fit in just like that. Because hopefully by now you know what we're going to be measuring. We're going to be measuring, because when seeds are doing respiration, right, we should be measuring how much CO2 or the displacement of that gas that's occurring. So what we're going to do, as directions say, and again I'm on third one, first thing we're going to place some absorbent cotton into the bottom of the first vial. So I'm just going to take a little piece of this, drop it in. We'll use something besides my stylus in class to get it down in there. I'm doing this off camera. But so we get on the bottom, we've got the absorbent cotton. Next, we've got to add some of our sodium hydroxide, which I have sitting over there with my pipetter. So let me go grab that before I try to add it. Alright, I'm back. I made a mistake. It's potassium hydroxide, not sodium hydroxide. But I got my cotton in there, place the absorbent cotton there, and then we're going to place, it says to saturate it, it says about 2 mils. Again, if you look on your pipetter, 1 full of these is 1 mil, so we're going to put in 2 full of this potassium hydroxide. So 1, and again, this is where we need the this is why we need the goggles here. So we'll definitely be wearing these. And 2. Now, if it looks like a lot of it's kind of sitting down there, you can always put a little more cotton in there just to make sure. We don't want it too saturated. So I'm going to shove a little bit more cotton in there to absorb any of that excess. So just like that, tap, 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 boom. Some hydroxide in there. And then we're going to add some non-absorbent, which I'm really not sure what the difference is. One absorbs and one doesn't, but I don't know the characteristics. I don't know the science behind it. But then we got our non-absorbent. So that's our first respometer set up. Once we do that, as the directions say here, we're now going to place our our germinating into the respometer. There they go. Try not to lose any of them, Mansoor. Like we just did. One fell in the water. It's alright. We do that. That's what your setup should be like. Absorbent, potassium hydroxide, non-absorbent peas. And then what I'm going to do, put this lid on and just let it sit off to the side. I'm going to go through and do the other 3. And alright, we're back. So I got my 3 respometers built. Here's the one with the beads, germinating, and our dormant. So not too difficult of a setup. So we'll kind of set those off to the side. Now if you look in the directions, it says optional if your instructor add the food coloring. I usually add the food coloring once I get them set in the water. So I'm going against the lab. Oh no, people at Carolina are going to be upset. So to get the tray set up, I take a piece of tape, almost hit me in the face, and I'll show you the setup. But I set it across one top part of the tray, And then I'm going to place respometers in there, and I'll show you all this. So here's first respometer, second respometer, and the third respometer, which is actually the control. Now, what you're going to notice, I'm going to show this to you in a second, I don't have enough water in here. Those respometers really need to be underwater. So what I'm going to do, I'm going to show you the setup, add some more water, and then we'll go from there. So just give me a second here to set this up, and then I'm going to... So again, here's the aerial view. I don't know if you can really tell, but I do not have enough water in there. So I've got the respometers sitting on that tape just so that they can calibrate. And going from top to bottom, we've got germinating, dormant, then the beads. So what I'm going to do, I'm going to fill this up with water, and then I'll come back to you. Alright, so I got some more water here. Went over to Miss Waggy's room, got a jug. And now I'm just going to add more water so that, as you can see the water coming out, so that my respometers will be completely covered. May have to adjust them a little bit. They are rolling around a little bit because of that white. Alright. So respometers are completely submerged in water, and what I can actually do, I just need to see the thermometer, so I'm going to move one of those white boards up so I can see the temperature. And so, I'm supposed to let these sit for 5 minutes. I'm going to show you what they look like here in a second. It seems each time I change the video, the line gets a little closer. I'm not sure if he's moving or if I'm moving the video. But in the water, gotta wait 5 minutes, allow time to equilibrium. So, alright. So I'm gonna start the timer. There's the timer. Alright, in 5 minutes I'll be back, and then we can submerge them. So, as the timer's going, there's one thing I do want to show you. I did add the red dye to the very tip of each of the respometers. And so as I'm looking at here, that dye should probably stay at the top until we submerge them. If the dye slides down the pipette, that means that there's probably a leak because it's going to be pulled down due to the difference in the into the pressure there due to the vacuum. So I've done that. Basically, all I've done, I'm just kind of give you a demonstration. So pretend this is the pipette. All I do is I barely touch the tip of the dye to it. I don't know if you can see that, but it just kind of should stay in the tip there. Okay. The red dye in the tips. What I'm going to do now is I'm going to gently submerge the respometers into the water. And so here we go. The respometers are set up. I started the timer. As you can see where the dye is. The directions tell you to record every 5 minutes for 20 minutes to the 0.01. Alright, so I got the lab set up. It's going to run for 20 minutes. What's probably going to happen is, being that we have a 2-hour block, this is a time when you're going to design your inquiry lab. Now I've got mung bean, barley, you can do peas again, and then I've got one other type of bean which I cannot think of off the top of my head right now. Barley, mung, peas, and I dumped them all over the floor. So the custodian's not going to be happy because I dropped a bag and they went everywhere. But anyways, during your inquiry, you are going to set up the same process. So you can look at factors such as temperature of the water maybe. You could maybe, if you want to see if some chemicals affect respiration of the peas, if you want to add an acid or a base. I don't care what you do, but it's an inquiry. So you're going to have all the same materials. I got alfalfa seeds, that's what it is. Alfalfa, it just came to me. Which, you know, Alfalfa, my Little Rascals, Alfalfa, he was the guy that had a little, okay, anyways. But anyways, so you're going to do your inquiry. So during this 20 minutes of prepping and running this one and seeing how bad I screwed it up, then we're going to let you run your own inquiry. And again, we'll be in our groups of 3. And so hopefully we'll see how things go. Remember, data, even if it's not what we expect, is still better than no data. And hopefully you guys understand that we're more concerned about the process. If we don't get every decimal point to the 12th place, we'll be alright. But trying to make you guys see the big picture here. Science is a process, not memorization. So I'm signing off from room 104. Leo's signing off too. He's going to go out and hunt some gazelles or some wildebeest out on the savannah later on today. Not really. But anyways, alright, sign off and have a good one.